Very good evening, children. Are you able to see me? Can you hear me, children? Are you able to see me? Can you hear me properly? Krishna Kumar. Yes, child. Somebody is in different login today. Okay, Ashini. Okay, ma. No problem. I thought some new student is there. Okay. We'll start the class. Today's class is just a continuation topic what we have learnt last class. Are you able to see the screen properly, children? Are you able to see the screen properly? Mahadev, just 1 minute, I'll also check once. Check. Okay, very good, children. So we'll continue the topic what we have learned last class. The topic is, last class we have learnt about the cell division process, and also we have learnt about cell cycle. In today's class I am going to explain about the mitosis and meiosis. The mitosis and meiosis. So what is the difference between the mitosis and meiosis? Just make a note. I am going to show you the, I am going to write the difference between mitosis and meiosis. Okay, children? So you can have a note with you. Mitosis and meiosis. Okay, children. So mitosis, it is a cell division which occurs in body cells. The body cells are known as somatic cells. Okay, children. The mitosis, it occurs in body cells. The body cells are known as somatic cells. Somatic cells. Similarly, the meiosis cell division, it occurs in sex cells. So that is the main difference between here. So the mitosis cell division, this occurs in the body cells. So body cells. So always the somatic cells that occurs in the body cells, it will be always the condition, it will be 2n. That is diploid conditions, it will be there. The condition, it will be 2n condition, that is diploid. Whereas the sex cells, it will be always n, that is haploid. The condition is haploid. So sex cells always, it will be diploid. That is the sex cells are always haploid in conditions, whereas the somatic cells are 2n. The 2n condition, we know it as diploid conditions. Diploid. Their condition is diploid. So that is the main difference between the meiosis and mitosis cell divisions. So the mitosis cell division, it occurs in the body cells. except the sperm and ovum, all cell division in our body, it is mitosis cell division. Whereas meiosis, this cell division, it occurs in the sex cells. The sex cells, you know, that is the egg or ovum, and in the case of male, it is the sperm. So the eggs and sperms, the cell division that takes place, we call it as meiosis cell divisions. Okay, children. So now we will move on to the concept of mitosis and meiosis. Mitosis, as I mentioned earlier, this is the cell division that occurs in the case of body cells, in body cells. And in the mitosis cell division, as I mentioned earlier, the cells are diploid in condition, 2n conditions. This diploid cells, it undergo the cell division where it forms two daughter cells. Both the daughter cells are again diploid in conditions. And here the multiplication of cells that occurs in a body, you know that the cells in a body keep on multiplying. That process we call it as growth and development. So that multiplication process, or even the development of zygote into a multicellular organisms, we know that we have started the life in a small zygote which is diploid. From that we become the multicellular organism. That division itself, it is called mitotic cell divisions. Whereas the formation of gametes, the sperm and ovum alone, that is meiosis process. The rest of the cell division in a body, it is mitosis cell divisions. So why mitosis is very, very important for us? Mitosis is very important for maintenance of cells in the body. There is something called programmed cell death in our body. Programmed cell death. What is programmed cell death? Cells are dying, cells are growing. You got it? That is called PCD, programmed cell death. In a body, cells are continuously, they are dying, cells are continuously replaced. That is called maintenance. For the maintenance, the mitosis is very important. Next is called growth. Growth from small to big. For example, at the time of birth, the weight probably it will be around 3 to 3.5 kg. Now if I ask you a weight, 40 kg, 50 kg, sometimes 60 kg, that is because of growth process. Similarly, repair. If there is an injury in the body, the cells get repaired. That repair, it is also because of mitosis process. And the continuity of the life, in the case of reproduction, in reproductive process, or the continuation of physiological process, the mitosis play an important role. And the mitosis, as I mentioned there, it usually occurs in somatic cells or vegetative cells, where a diploid parent divides and forms two diploid cells. Here the chromosomal number is maintained, remains the same. Here also it is diploid, here also it is diploid. That is about mitosis. Next is, in the mitosis cell divisions, the daughter cell, it exactly looks like the parent, both quantitatively and qualitatively. In both the cases, both quantitatively, qualitatively, this diploid cells, it will be exactly same like the parent. There is there is an increase in the number of cells, but the genetic makeup of the cell, it is same. Here one cell become two cells, but all the genetic makeup or the chromosomes, it remains the same. This mitosis cell division, it occurs in both plants and animals. In the case of plants, we can see that the meristematic cells, the root tips, the shoot tips, the lateral side, the intercalary meristems, in all those regions we can able to see the mitosis process. And if you want to study about the mitosis, the best suitable material is the root tip of onion. The root tip of onion, we can easily study the mitosis process. And this term mitosis, it was proposed by Fleming in the year 1882, and it was first observed in plants by Strasburger and animals by Fleming in the year 1879. So this is the mitosis process. In the mitosis, there are two types of mitosis takes place: closed mitosis and open mitosis. Can you all see this diagram? Just observe the diagram. This diagram, with the help of diagram, you can easily understand the process of closed mitosis and open mitosis. In the case of closed mitosis, the nuclear envelope, can you all see it here? This is the nucleic material, and the outer covering, what I am just showing it to you, that covering, it is called as the nuclear envelope. In the closed mitosis, the nuclear envelope, it is intact. Intact means it will not disappear. It will not disappear. Even the chromosomes move towards the pole, even in that time we can see that it remains the same. It remains the same. That is called closed mitosis. Here, the whole cell division process, it occurs within the nuclear envelope. You can see the nuclear envelope here also, here also. That's why it's called closed mitosis. This occurs mostly in the case of eukaryotic organisms, yeast and slim moles. Next is called the open mitosis. In the open mitosis, the nuclear envelope, which is here, this nuclear envelope, it will disappear and finally it will reappear. You got it? Nuclear envelope is here. This nuclear envelope, during cell division process, it will disappear, it will break down, and finally again at final stage again it is appearing there. So disappearing of nuclear membrane and reappearing of nuclear membrane takes place in open mitosis, whereas in closed mitosis, the nuclear envelope, it remains intact. Next we are going to talk about the stages of mitosis. Interphase, this is the starting phase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase, and these four phases, right? These four phases, it comes under karyokinesis. What is karyokinesis? Karyon means nucleus, kinesis means division. The nuclear division process takes place. Whereas the final separation of two cells, the separation of the cytoplasm, that is process is called cytokinesis. So altogether we can see there interphase, prophase, metaphase, anaphase, telophase. So 5 phases are there. So interphase, it is an intermediate phase between two successive mitoses. Got it? Between two successive mitoses, the interphase, it is a center phase. So we can calculate stages as prophase, metaphase, anaphase and telophase. This is about the stages of mitosis. Now I am going to explain about the prophase. The prophase. Okay. So now we can have a note and then we can write the points what I am just highlighting it to you. Okay. Just copy that thing alone so that it will be easy for you to understand the process. Okay children. Now just take a note, write the stages. I'll just show the stages to here. Just copy the stages. Okay, just write the stages. I'll tell you. Put mitosis, and then write prophase, metaphase, anaphase, telophase. 4 phases: prophase, metaphase, anaphase and telophase. These are the 4 phases that is there in mitosis cell divisions. Okay, first stage. Just observe the Just observe the picture here. Can you all see it here, children? This part, this is a typical cell. This is a diagram of a typical cell that undergoes the process called mitosis. Can you all see that? This is a cell, and this part, what I am just highlighting here, this is called as centrioles. This is called as centrioles. Okay, so here you can see that the membrane, what we are seeing it here, this membrane is called as nuclear membrane. And inside, what we are seeing it here, this is called as the chromatin threads. And also it has the nucleolus. So what are all present in interface? I am just explaining to you, children. The cell that is supposed to undergo the process called mitosis, it should have all these structures. It should have centrioles, the nuclear envelope, the chromatin threads, and nucleolus. Okay, have a note. 4 things it should have. One is called centrioles, nuclear membrane, chromatin threads, nucleolus. All these 4 structures are formed here. Now this cell, it will undergo a process called prophase. Prophase. This prophase, it is the longest phase in mitosis. So what will happen to the cell which is like this in prophase? What will happen? The chromatin threads. This is the chromatin threads. These chromatin threads condense and forms, and it become a chromosomes. First point, write it. The chromosomes, the chromatin threads condense and becomes chromosomes. That is what is happening in first phase. Okay. Next is the centrioles divide and move towards the poles. The nucleolar and nuclear membrane disappears. You got it? I just highlight the point here, children. Just make a note. What are things is happening here? First point, make a note. What is happening in prophase? What is happening in prophase? First point, the centrioles divide and move towards the poles. This is centrioles. The centrioles divides and move towards the poles. One here and one is on the opposite side. Next, point, the nuclear membrane and nucleolus disappears. Nuclear membrane and nucleolus disappears. Chromatin threads condense and become the chromosomes. So these are the points I want you to copy it. Okay, children, I'll write the points for you. In prophase. First point, what is happening there? Centrioles divide. Point number 2, nuclear membrane disappear. Then third point, nucleolus disappear. And fourth point, chromatin threads they condense and become chromosomes. These are the changes that takes place during cell division process. These are the changes that takes place during cell division process. Okay, children. Next, I am going to explain to you about what are the further changes takes place there. So now just have these 4 points in mind. This is what is happening during cell division process. Next, what will happen here? The chromatin fibers, right? These chromatin fibers, it becomes shorter and thicker. That's called condensation process, and they become chromosomes. And here, these structures what we are seeing, they are called chromatids. These chromatids, they joined at a particular point by a structure called centromere. Got it? Centromere. And finally, what will happen here during cell division process? A tetrad structure is formed. This is the tetrad structure with a centriole. This is what is formed during prophase stage. The chromatin threads condense and become the chromosomes. They form a structure called chromatids. Each chromosome consists of 2 sister chromatids. So this is one chromatid, this is another chromatid. These chromatids, they joined at a particular region. That region is called centromere. And the structure, a typical structure of chromosome is formed during prophase. Next, again, what I have told you, the nuclear membrane, the nucleolus disappear. And then this centrioles is there, right? These centrioles, they start producing astral fibers or spindle fibers. They start producing spindle fibers. So during this prophase, the Golgi complexes, endoplasmic reticulum, nucleolus and nuclear membrane, everything it will disappear. So finally, during this stage, what is formed here? During this stage, what is exactly formed here? We can see it here. A tetrad structure is formed. A tetrad structure. We can see that a chromosome is formed. This part, the center of the chromosome, we call it as centromere. The centromere is formed. A typical centromere structure is formed during cell division process. So at the end of prophase, we can see the chromatin threads, which was like this. These chromatin threads, they condense and become the chromosomes. So each chromosome, they have the center region, the region where the chromatids are attached. That is called a centromere. Centromere is formed. You suppose, just see that, children, the centromere is like that. At the side of the centromere, there is one more structure is there. That structure are called as kinetochore. The structure is called as kinetochores. Kinetochore is formed. So kinetochore, it is a structure that is towards the side of the centromere. Just now, I hope you can understand that at the end of the prophase, what is formed here. Again, I am going to repeat it for you, children. Just understand this. At the end of the prophase, We can see that the chromatin threads, they condense and become the chromosomes. They condense and become the chromosomes, and the chromosomes, the sister chromatids, they joined at the center region. That center region we call it as centromere. That region is called as centromere, and adjacent to the centromere, on the side of the centromere, the kinetochores are formed. The kinetochore are formed. This kinetochore, it is very important for cell division. Why? I'm going to explain it to you. If you take a cell, the centrioles, it will be here. Centrioles are here. During cell division, what will happen? Just assume that this is a centriole. During cell division process, the centrioles, they start producing spindle fibers. This spindle fibers attaches to the kinetochore like this. They'll get attaches on both side of the centrioles. They start producing spindle fibers. Those spindle fibers attaches to the kinetochore. So during cell division, what will happen? This kinetochore, this spindle fibers that are formed from the centriole, this part it is called a centriole. This centriole, they produce spindle fibers. The spindle fibers that are formed from centriole attaches to the kinetochore. During nuclear division, that is karyokinesis process, what will happen? The spindle fibers, it will move like this. It will contract. When it contracts, what will happen? The nucleus divides into two. This is called karyokinesis process. Okay, children, just now I'm going to summarize the prophase phase. I want you to understand that properly. What is happening during prophase? At the end of prophase, what will happen? The cell that is like this interphase, it will become like this. So what are the changes takes place there? The changes takes place, the chromatin threads condense and become the chromosomes. The chromosomes attaches at a particular point, that is called centromere is formed. At the side of the centromere, kinetochores are formed. Two kinetochores are formed, and the nucleolus, the nuclear membrane, Golgi complex, endoplasmic reticulum, everything it will disappear at the end of prophase. And the centrioles, they start producing astral rays or spindle fibers. That is what is happening in prophase. The next stage, it is called metaphase. In the metaphase, what is happening here? In the previous structure, you can see that the chromosomes are scattered in the cytoplasm. Can you all see that? The chromosomes are scattered in the cytoplasm. Whereas here, what will happen? The chromosomes are arranged in a central plate. This is called metaphase plate or equatorial plate. So how it is arranged? It is arranged by the spindle fibers. I have told you already about the kinetochore. Can you see that? This is the kinetochore structure. This part is called centromere, and there are two kinetochores are there. The spindle fibers are microtubules that are formed from the centrioles. It attaches to the kinetochore, and this only it arrange all the chromosomes at the equatorial plate. So the equator region, they are arranged in a straight line. In the metaphase plate, they are arranged in a straight line with the help of the spindle fibers that are formed from the centriole. This is called centriole. The centriole start producing spindle fibers. Those spindle fibers attaches to the kinetochore of the chromosomes and arrange all the chromosomes in a metaphase plate, in a equatorial plate at the center. The centrioles, they produce spindle fiber. Those spindle fibers are made up of a special protein called tubulin. Now, this is what is happening in the metaphase stage. In the metaphase stage, what will happen? The chromosomes are arranged exactly at the equator. Are they arranged in the metaphase plate or the center with the help of the spindle fibers which are formed from the centrioles. Next is anaphase. So before anaphase, I want to clarify all your doubts. Do you have any doubt, children? If you have any doubt, you can post your doubts now. I'm there to clarify your doubts. Any doubt, children? Deepak, Vashini, Kadiravan, Madesh, Madhe, Krishna, Charvi. Do you have any doubt? Charvi, Kadiravan புரிஞ்சுதா எல்லாமே உங்களுக்கு? தீபக் புரிஞ்சுதா? Okay, very good children. So at the end of the metaphase, all the cells are arranged at the equatorial plate. That's what I want you to understand. Next, you can see it here what is happening. The next stage, anaphase stage. The previous stage, you can see that the cell, it will be like this. What happened here? On the metaphase plate, all the chromosomes are arranged, spindle fibers are attached to the kinetochore. To the kinetochore, spindle fibers are attached. Now what will happen here? During anaphase, the centrioles, it will shorten the spindle fiber. It will shorten the spindle fiber. It just pulls like this, just like a tug of war, right? Two people, they'll try to pull on opposite sides. Like that, what will happen? The spindle fibers that are attached to the kinetochore, it starts shortening. Because of that, what will happen? Each chromosomes which are lined here, they get separated like this. So now these chromosomes, it again split and become chromatids. And each chromatids, the sister chromatids, it move towards the poles. During this time, what will happen? The center of the chromosome, that is called centromere. The centromere split longitudinally into two. One half of the centromere, it moves with the sister chromatid, one with other sister chromatids. Similarly, what will happen? When they divide, the kinetochore also divides equally. The centriole, the centromere, the chromatids, the kinetochore, it divides into two equal half and move towards the poles. Like this, it will be there, and finally it will become like this. This process of separation of the chromosomes, we call it as karyokinesis. Karyokinesis process. Here, equal amount of genomes. What is genome? The complete set of genes. Before separation, it will get replicated. It will be in two numbers. When they separate, it divides equally. This is what is happening during anaphase stage. The final phase, it is called telophase. In telophase stage, what will happen? The chromatids which are moving towards the pole, it reaches the pole, opposite pole, and the mitotic spindle that is formed here from the centrioles, it will disappear. And then the centrioles which was lost, which disappeared during prophase, it will reappear. And then the nuclear membrane, it will reappear. You got it? Nucleolus reappear, nuclear membrane reappear, Golgi body, endoplasmic reticulum, everything it will reappear. So at the end of the telophase, the genetic material is completely separated. That is called karyokinesis process is over. Now we can see that the division of cytoplasm starts. And during this process, what will happen? The nucleus, the chromatrix which was like this, it will decondense and become chromatin threads. Nucleolus reappears, nuclear membrane reappears, and the chromatids, it again decondense and become chromatin threads. So at the end of this telophase, we can see that two daughter cells are formed. These two daughter cells are exactly the same like the interphase cells. It will be same like the interphase cells, where you can see the centrioles, it will be there, the nuclear membrane, the nucleolus, endoplasmic reticulum, Golgi body, everything it will be there. And now the cell plate is formed like this. It will divide. Once they divide, there are two ways by which the cell plates are formed that I am going to explain it to you. So now what will happen? The plate is formed here. This plate is called cell plate. That cell plate, it helps in division of the this is called cell plate. This cell plate helps in division of the two daughter cells in plant cells and helps in formation of cell wall. That's what I am going to explain to you later. Here the cell plate is formed between the two daughter cells and reconstruction of cell wall takes place in the plant cell, whereas in the animal cell, just like that, the cell divides. At the end of this telophase stage, we can see that two daughter cells are formed. This is what the cell division process. So here you can see that the one cell that enters into interphase, it will undergo a lot of changes in prophase, and then it will arrange them in order in the metaphase. They started moving to the poles towards the poles in anaphase, and finally in telophase they form the structure. The lost structures are again formed and complete transition of cell takes place where two daughter cells are formed. The two daughter cells are exactly same in like interphase cells. And now I am going to explain to you about the animal cytokinesis process. So far what we have seen is called karyokinesis, division of nucleus. Now the division of cytoplasm we are going to talk about. In animal cytokinesis process, the spindle cells which are formed from the centrioles, they form a ring-like structure. At the mid, they will form a dense fibrous and a ring-like structure that is called contractile ring. And this structure that is formed at the center induces the formation of cell membrane to invaginate. Once the contractile ring is formed, it will induce the outer cell membrane to invaginate. Invaginate means it moves inside. So now they started moving inside. In both the sides, it started moving inside. They'll move it like this slowly, and then it will form the structure. So this method it is also called as furrow method. Furrow method, where once this process is completed, two daughter cells are formed. So during this process, the cell organelles like mitochondria, ribosomes, Golgi body, endoplasmic reticulum, ribosomes, everything are completely formed and distributed equally to the two daughter cells. This is called animal cytokinesis. Whereas in the case of plant cytokinesis process, it is cell plate method. Here, as I told, that the spindle fibers, it forms a dense fibrous structure and then it helps in formation of the cell membrane. Here, the cell wall has to be formed. Here, the cell wall, it is formed by a small vesicles of Golgi complex. The small vesicles of Golgi complex called Golgi vesicles, it attaches, it is arranged at the center of the cell. That's called equator region. And finally, what will happen? All these small vesicle, Golgi vesicle, they'll fuse together and they form a two sheets. They'll form a structure called cell plate, or middle lamellae are formed. Then what will happen? It grows centrifugally. And then it grows like this from center to outer, and finally it comes in contact with the outer membrane. And finally what will happen? They'll form a typical structure like this. And this formation structure, it includes cellulose. It requires cellulose, hemicellulose, pectin, and all the structures. It requires certain minerals to form this typical cell plate structures. Or formation of the cell wall, it requires cellulose, hemicellulose, pectin, and all of them get deposited on the cell plate, and finally we can see that two daughter cells are formed. This is called plant cytokinesis. Now, I just clear your doubts and then I'll explain to you. Kadiravan, mitotic spindle pulls the sister chromatids separate. No, yes, you are right. The mitotic spindle only contracts. When the mitotic spindle contracts, the chromosome divides. The centromere divides, the chromatids divides, and the kinetochore divides. You got it? Vashini, how does the daughter cell remain identical? The spindle fiber randomly pulls different chromatids. See here, they are exactly attaching to the one side of chromatids. Just 1 minute, I'll just explain to you. I'll just change the slide so that you can understand it better. Just Vashini, just observe it here. This is a typical structure of a chromosomes. So this point, this is called as centromere, and this part here only what is there? The kinetochores are here. This is the region where the spindle fibers are formed, and from the centriole the spindle fibers are formed. The spindle fiber not only at the kinetochore, they are also present like this. Astral fibers. Okay, so now you can see that the center part, centromere, and here. Now the centrioles, it will contract the spindle fibers. The spindle fibers are now contracted. It shortens the spindle fiber in this direction. Once it will pull that, here exactly it will divide. The center it will divide. When the center divide, what will happen? Two equal structures are formed. So at the center it will divide like this. So you can see it here, exactly at the center it will divide. Here in the center region it will divide at middle. So that what will happen? This side you can see this chromatid, it will move to this side, this pole. This is one pole, P1. I'm just marking it as P2. So from here the centrioles divide, the centromere divides, and the kinetochore moves, kinetochore, and this centromere, this part, this kinetochore, and this chromatid, everything it will move towards this side. So equal division it will be there. That is the reason why all these spindle fibers here, everything is completely attaches to the chromosomes so that it will divide equally. That is the reason why in mitosis equal division is there. Whereas in the case of amitosis process, amitosis, there is no equal division like this. There is no equal division like this. Equal division will not be there. The equal division only it occurs in amitosis, occurs in mitosis process, not in amitosis process. Clear, ma? Are you all clear with that, children? Others, do you have any doubt? Vashini and Kadiravan, are you all clear with the concept? Others, do you have any doubt, children? Okay, so next I am going to explain to you about significance of mitosis. Significance of mitosis. In the mitosis process, exact copy of parental cell is formed. Qualitatively, quantitatively similar. That means genetically identical daughter cells are formed in mitosis. That is genetic stability. The daughter cells genetically identical to the parent, and growth. The mitosis helps in growth of the cell. One cell, the zygote, it develops into multicellular organisms. How this is possible? This is possible only because of increase in the number of cells. Repair of tissues. The damaged cells are repaired with the help of mitosis process. Whereas the lower organisms like yeast and amoeba, the reproduction itself, it is by a process called mitosis, asexual reproduction. That is the reason why in asexual reproductions, all the daughter cells that are formed, they are exactly similar to the parent. Morphologically, genetically, physiologically, behavioral wise, all characters are same. Next is in flowering plants, the bulbs, corms, tubers, rhizomes, runners, everything it is formed by mitotic divisions. Similarly, the regeneration process, the formation of new arm, regeneration process. In the case of starfish, it is again mitosis process. Similarly, in the case of planaria, it is again mitosis process. And in plant tissue culture process, they are using the process is mitosis process. So the mitosis process, in a very short period of time, we can produce lot of offsprings. That is the significance of mitosis process. Next we are going to talk about meiosis. The meiosis process, I want you to draw the diagrams here. Just write this. I'll give you 5 minutes time. Just copy this, and also okay, just copy this alone. I'll come and explain the process after 5 minutes. Meanwhile, if you have any doubt, you can post your doubts, children. Welcome back, children. I hope you could have completed the topic, the thing. Now I'm going to explain to you about the meiosis cell division. Next I'm going to explain to you about the meiosis cell division. Have you all done it, children? Have you all done it? Have you all done it? Yes. Okay. Very good. Very good. Okay. The next topic is meiosis cell division. This meiosis cell division, as I mentioned earlier, it occurs in sex cells. It occurs in sex cells, or the formation of gamete. The formation of gamete. The formation of gamete. There is a process that is called gametogenesis. Gametogenesis. That means formation of gametes are known as gametogenesis process. For that gametogenesis process, meiosis is the only cell division process, and this was first discovered by Oscar Hedwig in sea urchin eggs. Similarly, Edward von Benden, it described the level of chromosomes in Ascariasis eggs in the year 1883. The production of gametes, because we know that all the gametes are haploid structure, they are haploid. So in the production of gamete, in the meiosis process, it is the only process by which the gametes are formed. And the gametes like ovum and egg, they are formed by a process called meiosis, and during reproduction, this meiosis haploid cells, they combine, join together, and they become zygote, diploid cell. This diploid cell, it undergo a process called mitosis, and they develop into the multicellular organisms. And here next we are going to talk about the cell division, overall cell division process. This meiosis cell division, it occurs in two phases. One is called meiosis 1 and meiosis 2. The meiosis 1 and meiosis 2. In the meiosis 1, it has 4 phases. Similarly, the meiosis 2, it has 4 phases. In the meiosis cell division, the longest phase, it is prophase 1. The longest phase is prophase 1. The prophase 1, it has 5 stages: leptotin, zygotin, paketin, diplotin, diakinesis. And after diakinesis only metaphase 1. We all know what will happen in prophase 1. We already learnt about mitosis. What will happen in metaphase? What will happen in anaphase? What will happen in telophase? That just have that in mind so that you can easily understand the meiosis process. So meiosis, it is called double division. Why? Because 2 times the cells divide. 2 times the cells divide. Metaphase 1, and then like prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, telophase 1 and telophase 2. So like that 4 cells are formed, whereas in mitosis only 2 daughter cells are formed. The diploid cell, first division is called interphase, and after interphase prophase 1, metaphase 1, anaphase 1, telophase 1. After telophase prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis, and then 4 haploid cells are formed. So here the same diploid cell, it undergoes the process called cell division and forms 4 haploid cells. So at the end of the meiosis process, 4 haploid cells are formed. And here the meiosis, the cells that undergo meiosis are called myocytes. There are two myocytes, are the spermatocytes in the case of males, oocytes in the case of females. This is a continuous process, sequential process, meiosis 1 followed by meiosis 2. And here in meiosis, the pairing of homologous chromosomes takes place that results in recombination. Whereas in mitosis cell division, there is no recombination takes place. The recombination between the chromosomes, it takes place only during meiosis process. At the end of meiosis, 4 haploid cells are formed, and the transitional phase between meiosis 1 and meiosis 2, it is called interkinesis. Interkinesis or intrameiotic interphase. So this is about the clear idea about meiosis. Now we'll move on to the phases. First, prophase 1. As I mentioned earlier, the prophase 1 is the longest phase of meiotic division, longest phase. The cell that enters into prophase 1, same like interphase. In the prophase of meiosis, in the mitosis, what will happen? The chromatin threads condense and become the chromosomes. Similar, it will take place here, but everything it occurs in stages. Here also the DNA coils tighter, and the individual chromosomes are visible like this. And here the sex chromosomes, because this occurs in sex cells, the sex chromosomes are heterochromatic bodies, different colors, different color. Chroma means color, hetero means different. Different color bodies it will be. That's why it is shown in 2 different colors here. There are 5 subphases in prophase 1: leptotin, zygotin, pachytin, diplotin, and diakinesis. So you can see it here. The first stage, leptotin. Leptos means tin. Tania means band. So here you can see that the nucleus are chromatin threads. It increased in size, and chromosome, it will appear like a thread like structure, a typical thread like structure, leptotin thread like structure, that's called leptotin. So the chromosomes, the chromatin threads, now it is clearly visible like a thread like structure, that is called leptotin. And you can see that these chromosomes, they have a bead like structure. That bead like structure is called centromere. The centromere that divides the chromosomes into 2 chromatids. Next is zygotin. Now see the chromosomes here. There are heterochromatids, 2 different colors it is there. Now what will happen here? The 2 sets of chromosomes, this is one set, the other set, 2 sets of chromosomes. The number is diploid conditions. These chromosomes are called as homologous chromosomes. If similar type is there, it's called homologous chromosomes. The homologous chromosomes, it resembles in each other. For example, here you can see that one is short, the other one is long. Here also one is short, the other one is long. So what will happen here? The pairing of chromosomes takes place based on the structure. So you can see that 2 short and 2 long chromosomes and centromeres. Like that, it will arrange themselves in a position of the centromere. And similarly, what will happen? The 2 homologous chromosomes, the similar chromosomes, they get attached laterally in sides. They'll get attached. They'll get attached laterally. The sides, they will get attached. like this. They will attached, and during attachment they form a complex called synaptonemal complex. Synaptonemal complex. This synaptonemal complex that is formed between the two chromosomes, it is because of a nucleoprotein, and this process it is also called as synapsis. That's called attachment of homologous chromosomes. Attachment of homologous chromosomes, that is called synapsis. The synapsis is formed because of synaptonemal complex. Now what is happening in leptotin? Thread-like chromosomes are formed. Here pairing of chromosomes takes place. That is called zygotin. Next is called patchotin. The patchotin, what will happen? The crossing over between two non-sister chromatids, it will take place. The two non-sister chromatids, the crossing over takes place. How the crossing over takes place? The bivalent chromosomes, it will come together like this nearby, and the exchange of segment takes place here. So these two are sister chromatids and these two are non-sister chromatids. This is sister chromatids, this is our sister chromatids, but these two are non-sister chromatids. The two chromatids belong to the same chromosome, we call it sister chromatids. Different chromosome, non-sister chromatids. Both the non-sister chromatids, it comes together. It will come together like this. And then crossing over takes place. The two non-sister chromatids comes together and then the exchange of segment takes place here. After that, it will become like this. So the meiosis is the only cell division where the crossing over of chromatid segment takes place. The crossing over of chromatid between the non-sister chromatids, it is called crossing over. The exchange of segment is called crossing over. For that crossing over, it is an enzyme mediated process. A lot of enzymes are involved in that. During the zygotin, the synapsis takes place. During patchotin, crossing over takes place. And this recombination, the crossing over between the two non-sister chromatids, lot of enzymes are involved in that. That is called as recombinase. Recombinase enzymes. The recombinase enzymes, it includes exonucleases, endonucleases, unwindase, or protein. These are all the enzymes that are involved in exchanging of segment between the non-sister chromatids takes place. The next stage, it is called diplotin stage. In the diplotin stage, what will happen here? The homologous chromosomes separate, except they will be first, it will be like this. The separation, it will be like this. Except one part, the rest of the chromosomes, it gets separated. Previously it was like this, and now except this part, the other parts get separated. You can clearly see the chromosomal structure during diplotin stage. Here you can see that the point of attachment between the two non-sister chromatids, it is called chiasmata. Previously both of them, they joined together like this, closed attachment. Now the attachment, it is only at a specific point. That specific point we call it as chiasmata. Chiasmata. Next process, diakinesis. Chiasmata. What is chiasmata? The attachment point, right? Now it gets separated. That process is called terminalization process. That is called diakinesis process. During diakinesis, what will happen? During prophase of mitosis, what are all things happened? Can anyone tell? What all happened during the end of prophase? Can anyone tell? Chiasmata, it is a point of attachment. Centromere is the center of the chromosomes. Two chromatids joined at a particular point. That point is called a centromere. Just it looks like a bead like structure, the attachment, that's called centromere. Chiasmata, it is point of attachment. இங்க நல்லா கவனிங்க வஷினி, ரெண்டு குரோமோசோம்ஸ். ஒரே நிமிஷம் I'll just explain it to you. See here children, first it looks like a thread like structure. This is called leptotin, and the leptotin you can see that there will be a dot, that this is called the centromere. Inga ஒரு dot இருக்கும், the dot பேர் the centromere. And finally what will happen, this they become tetrad structure. It will become like this. This point, that point, it is called a centromere. This point is there, this is called centromere. And again I am drawing one more here. Okay, so now two different chromosomes I have drawn it here. Can you see it here? Two different chromosomes I have drawn it here. This chromosomes, this is sister chromosome, this is another sister chromosome. Got it? This is sister chromosomes, but these two are non-sister chromosomes. This is one sister chromosome, this is another sister chromosomes, but these both are non-sister chromosomes. They are non-sister chromosomes. Now what will happen? The pairing takes place. Where exactly pairing takes place? The pairing takes place between the non-sister chromatids, like this. Got it? Now they'll come close together here. This process of coming together and attaching to that, attaching here like this, they will attach. This process, it is called the synapsis. This process called the synapsis. And then what will happen? The exchange of segment takes place here, from here to here, and similarly from here to here. Can you observe it here? The exchange of segment takes place here. The exchange of segment takes place here, and after exchange of segment, what will happen? The chromosomes, it will look alike. It will be like this. After exchange, one half of the chromatid it will be like this. Then the next half, it will be like this. After exchange. Similarly here, after exchange, it will be like this. But still what will happen, this part, it is attached here like this. So this exchange we call it as crossing over. The exchange we call it as crossing over. After crossing over also, what will happen here? The attachment, it will be here. There is an attachment here. Can you see it here? This point, there is an attachment. This point of attachment, it is called as chiasmata. The point of attachment is called as chiasmata. And during diakinesis only what will happen? The separation of this takes place. Till then, the tip like this, they are attached at one point. This point of attachment is called as chiasmata. Whereas, if you take a chromosome, the center of the chromosome, this part, it is called as centromere. Centromere is different from chiasmata. Here, synapsis process takes place. I hope all of you remember that, right? What are the process takes place here? Synapsis. Synapsis takes place. After synapsis, crossing over takes place. Remember, children? After crossing over, they get separate but attached to one particular point, that is called as chiasmata. And after chiasmata, and what will happen? The terminalization process takes place. Terminalization process means separation. The separation process takes place. This is all about the cell division process. Okay. Now, before the doubt clearing, I have asked you few things, right? What are the changes that takes place in prophase? Very good. Most of your answer is correctly. Formation of spindle fibers. Madhav Krishna, formation of spindle fibers. The spindle fiber formation, it occurs from centrioles. So for that, centrioles has to divide. Yes, Charvi, you have told that answer. All organelles disappear. Very good. Nucleolus disappear. Nuclear membrane disappears. Chromatin condense and become the chromosomes at the end. Now we can see that in prophase, after all the process, the chromosome is, the chromatin threads condense and become the chromosomes. Centrioles split into two and move towards the poles. So all these process takes place there. So at the end you can see that all these structures are formed. The centrioles move towards the pole and the nuclear membrane and nucleolus disappear, and the chiasmata, it gets separated. It's called terminalization process takes place. Next is metaphase stage. In metaphase you can see there the nucleolus, nuclear membrane, everything will disappear. The centrioles, it start producing spindle fiber. The same process metaphase in mitosis. It arranges them in the equatorial plate. And then the centrioles start producing spindle fiber those spindle fiber attaches to the kinetochores. And now what will happen? Metaphase stage. What is the next stage? Anaphase stage. In anaphase stage, what will happen? It will contract. The homologous chromosomes are separated now. It is like this, but a small change here, what will happen? The whole chromosomes, it will move towards this side. This whole, it comes towards this side. You can see it here. It will move like that. The homologous chromosomes are separated from each other due to shortening of the spindle fibers by the centrioles. Now, during this process, during this anaphase 1, centromere is undivided, kinetochore is undivided, chromatids are undivided. Just like that, it moves towards the poles. The whole homologous chromosomes, it will move towards the poles. These two chromosomes, one is maternal, the other one is paternal. What is maternal? That is from the father. Maternal means from the mother. Paternal means from the father. So here you can see that the whole structure, chromosomes, it has the chromatids, it has centromere, it has kinetochore, all the chromatids, it will be there. Next is telophase stage. In the telophase stage, what will happen? The same telophase in mitosis, understand that. They will move towards the pole and all the structures, it will reappear. What will reappear? Nucleolus reappear, nuclear membrane reappears, and the cell organelles reappears. Spindle fibers disappears. So everything, it will be formed. So this is what is happening during telophase 1. Now at the end of telophase, you can see that the cell division takes place and they form two daughter cells. These two daughter cells are still diploid conditions. And now after telophase 1, the cell enters into interkinesis. Interkinesis is very short-lived period. After interkinesis, the two daughter cells that are formed at the end of telophase 1, it will move to meiosis 2. It will move to meiosis 2. So from meiosis 2, there are few slides are there. Only around 8 slides are there. I'll do it in next class. So till now, do you have any doubt? I will clarify a doubt, children. No doubts. Have you understood the concept, children, all of you? Okay, please go through the textbook now itself. Don't delay, children. Now itself go through it and understand what is there. If you have any doubt, please ask me today itself. 8 o'clock, from 8 to 10 or 11 o'clock, you feel free to ask any doubt. But do it by today itself so that you can finish up the topic by today itself. You got it, children? Thank you very much, children. Thank you very much. We'll see you next class. Thank you.